But I think there are a few things that probably we need to kind of outline to everyone about cancer, since probably we're not everyone involved into cancer research. So, plastic tissue is in principle heterogeneous, has differences in terms of its microstructure, heterogeneity. There is unregulated balance of growth factors, enzymes, proteins, etc. Interaction between healthy cells as well as cancer-associated cells has been disturbed, as well as the function of the lymphatic vessels and blood vessels is known as it is a healthy tissue. At the bottom of the slide, I have several electron microscopy images, images, histology images, just showing the differences between healthy and tumor vessels, and so on. Now, the idea here is for cancer biologists and for engineers, how can we understand about what's causing those defects? We're focusing primarily on the first few slides on the blood vessels, and that's because blood vessels not only support healthy tissue providing nutrients, oxygen, but also blood vessels they are used for drug delivery, right? And in order to understand, say, how cancer develops, biologists, people coming from the medical field, they've been using for several decades in vitro models as well as in vivo models. So in order to understand physiology of cancer. However, there are models, so they are reductionist approaches of cancer, so they are simplified approaches. And however, there is this direction in bringing together not only biology but also engineering, physics, pure science together. And from that angle, Silico models, i.e. mathematical computational models, are being extensively used in the past couple of decades to understand cancer, how to aggregate information coming from different sources of data, like preclinical models, and imaging, in order to test new hypotheses, develop novel procedures for the system, certification diagnosis. And I guess the overall aim of Silico modeling is to use it as a little tool towards using animal testing, using the costs associated with the trials on humans. Now, and that's where we come from. What's our focus? What we aim in our group is we wish to build in Silico models and Silico tools, we call them models, in short, that encompass major mechanisms of tumors. Primarily, we are focusing on solid tumors. We're not focusing on tumors for one. And also use those models to effectively describe the different skills, which is one of the limitations of in vitro experience. And we want to, of course, use those models to simulate chemotherapeutic strategies, monotherapy, combined therapy, and so on, therapy, as well as use those as means of diagnosis and prognosis. Now, in terms of modeling, the list of models, mathematical models in biology is quite vast. Several papers, review articles, including that Miriam did a couple of years ago. The models are distinguished based on the scale that they try to describe, like coming from the bottom, this cartoon on the left, the atomistic level, up to the full body or organ scale. However, in my group, we focus primarily on the tissue down to the cell scale level. And we model using well-established finite element model techniques, as well as based modeling. In this talk, I will focus primarily on the first part of the finite element applications. So some simulations on the first, so starting by showing some demos, some simulations and of models that try to replicate what happens on an in vivo animal model. This kind of work that took quite a few years back while I was at UCL. So the initiative was to build this model that describes the tumor, the tissue, the mechanics in terms of as you can see here, the vascular. So all those different components that consist of the major, let's say, important components of the growing tumor. So we started by simulating tissue growth, capillary modeling. So we have this component, let's say, different modules describe the dynamics of the tube, of the blood vessels, as well as the vessels recruited by the tumor, as well as different modules describing the balance of biochemical fluids, like enzymes, proteins, oxygen, nutrients, and so on. We describe effectively the way that vessels sprout, the elongate, the form branches, the shape anastomosis, etc. And we use mathematical rules to describe that. Now, in this one of the top left, we have two different kind of ways of describing the geometry. So we have the final mesh on the left, which is tissue, where we kind of solve the different PDEs of the cytokines, the cellular matrices, as I mentioned before. So having four, let's say, PDEs in that sense, as well as a different, let's say, mesh, different discretization, describing the blood vessels. So those were two non-conforming meshes coupled together. But apart from that, we also had the mechanics, the solid mechanics and the fluid mechanics. So the solid mechanics in terms of the fluid mechanics starting off in terms of interstitial fluid flow, so flow in the extracellular matrix, flow in the blood vessels, as well as the transport in the vessels of the ECM and vice versa. And the solid mechanics, i.e. the mechanics from the deformations, stresses, strains, all those exotic things associated with the growing tumor, and as well as the structural effect of the not well known to you, but while tumor grows, tends to compress, to put some pressure to the vessels, as well as to the lymphatic system, and it causes some abnormalities. And we effectively see model that. So the vessels do deform. Now, this slide that I'm trying to just briefly demonstrate the validation process. So we validated the model. The model that we sum up very quickly describe the growing tumor, the mechanics of it, blood flow, chemicals, as well as the dynamics of vascular remodeling and sprouting. Okay. And we want to validate this is simulators some way produces vascular networks that are indeed true. So we compared the in silico simulated sort of vascular group, 3D vascular group, with those without of in vivo data. So those are optical frequency domain imaging scans, mouse, and we compare different methods. One was the vascular test, which is the bottom plot. Another one was the fractal dimensional of the vascular tree. And we compared the two different, let's say, metrics between the vivo and the in silico. Other metrics were like the fluid mechanics perspective, the fluid pressure. Other metrics were also the volume. Now, and we use those information all coupled together in order to make a confirmation that the silico does produce realistic results, and we also, tested the silico in terms of the mechanism. So that was one of the reasons, and that's actually one of the selling points of mechanistic models. compared to machine learning models. In mechanistic models, you describe mechanisms, which you then subsequently test, and you confirm whether mechanisms introduced into the models really make sense. Do agree with your in vivo, well, with your experimental evidence. Okay? And then a few years later, led by Miriam, we have more details. So we continue to have more features. So we enhance the this model such that explicitly includes healthy and tumor cell density variations. And we also describe effectively different phenotypic behavior of the cells, such as cancer cells can be proliferative, reactive, in other words, hypoxic, necrotic, as well as we also explicitly model the extracellular matrix modeling with respect to the growth of the cells, etc. Yeah, I reserve for the details on the paper that was published last year. But to be honest, it was kind of a very long list of PDEs and equations that we had to solve, together with next parameter that include those drug delivery.
So I think we can prepare questions now or after?
Now,
I have a question because I don't know this area very well. So are these like the questions that we see, are they basically phenomenological models that just
exactly. So the ones, for example, I showed currently and that describe the transport of drugs or the balance of drugs are based on KBD well-established models. Okay? Tuned on this particular case in order to describe, for example, the extravasation of drugs within the endothelium wall, as well as the absorption of the drugs in the lymphatic systems. Those are, let's say, two very dedicated mechanisms introduced into the model, but these are quite well established.
And the parameters,
yeah,
the parameters are typically from experimental measurements, or we just like tune the parameters to fit experimental data?
A combination of the three, meaning tuning, well, adapting from vivo, like I showed in the previous slide, adapting for vitro. So some tests have been done by others on in vitro, let's say, settings, as well as the third part, which is adapting from our side. Meaning that parameters tune or at least others managed to converge to realistic, say, value ranges were tailored to their models. So we have to adapt those parameters ourselves. So yeah, so different compartments, solid mechanics, fluid mechanics, vascular modeling, balance of chemical cues, and drug delivery all together under a single, let's say, framework in C++. We've been developing over the years. And then we, of course, want to take a few steps further, and apart from simulating tumor growth in angiogenesis, want to also simulate drug delivery, which is the real thing for us. So different scenarios starting from the top to bottom, single dose intravenous injection, and then different scenarios as we go along, where we try to simulate vascular density reduction. This is in effect the outcome of anti-angiogenic treatment. So this is what's been done on patients. And we want to simulate that on a mouse model. So what we've seen so far were simulations on mouse model. We're trying to simulate that again on a mouse model with anti-angiogenic drug by using different drugs that kind of affect the wall of the blood vessels, that are some call it vascular normalization. Other techniques like autonomic chemotherapy, like giving smaller doses instead of a big one, a single big one, as well as combined therapeutic treatments. So this, yeah.
Can you play with the process
What those field vessels?
of the vessel. Come out.
Yeah, absolutely. This was, I don't know if that, sorry. This is something we try to prove with this very particular animation comparison where we activated or deactivated some mechanisms and we observed letting some mechanisms like haptotaxis or the countertaxis. Sorry, I didn't go into the fine details, but those in effect allow for those observations to take place. So by triggering haptotaxis, yes, vessels try to follow the path, the least, let's say, extracellular matrix density, so that the endothelial cells try to probe the matrix in a way so that they form networks that kind of have the least resistance. And that's because of the tumor that while it grows, it kind of accumulates some force. And the vessels start to escape that. And this was done to but also there's the other aspect of the vascular remodeling. Yeah. I'm sorry that I'm not covering all the details, but I'll try to make it as short as possible. But then after having this model validated, we run those first single bolus injection drugs. And we want to use those simulations in order to understand about how different parameters like, let's say, affinity rate of the drug with respect to the pore size of the vessels, which is not, let's say, healthy in the tumor microenvironment, how this affects the amount of drug delivered to the tumor, how this in principle affects the body. So the larger the cytotoxic drug, of course, the larger the effect it will have on progressing the tumor. But this comes, from different perspectives, so from different parameters. And so have on the bottom left bubble that the time of drug administration affects tumor regression, but for low affinity, not that pronounced in high affinity drugs. So having a drug that binds very fast to the cancer cells, that does not necessarily mean that it will have the best effect. This is just an example. You see over on the right some animations in the control and treated scenarios, and those contours indicating the amount of drug bound to the tumor. But we also compared other metrics like this delta max parameter that gives the estimate of the short distance between the extracellular matrix and the blood vessels, and that because in healthy tissue, this delta max is almost uniform. The vascular network is uniform in the but in the tumor is not accepted. And we want to inspect in time how this plays out in the control, which is well, sorry, it's not very evident, but it's those black solid lines over here. And with respect to the different, let's say, types of administration, and with respect to the different affinity rates, and with respect to the different pore size of the tumor. So you see different parameters, different simulations, say illustrating a few plots. I'm so sorry it's too complicated, but one of the things that we observe that pore size, right here, this bubble affects the vascular texture normalization for low affinity, but not for high affinity. As a toxic drug. That was a very interesting finding, as well as through those heat maps that indicate, let's say, the value of the delta max, which is the shortest distance, the distance is in the vessels, as well as this fractional parameter. It indicated those windows as bottom that can be used as a means for a follow-up, which is exactly what I'm going to show there. Because in effect, you would like to regress the tumor and then use some other drug to make some other kind of normalization so as to reduce the risks of relapse of the cardiac.
So how does this data compare to experiment?
Yeah. There's no such data like the two plots I showed on the right. No way. But plots like plots on the right, you can have experimental data like of the kind volume of time. Volume of time. Yeah, but those plots do not represent tumor volume of time. They represent those geometric features post-process after the end of the simulation that indicate the distance between the vessels. So the gap between the vessels, which may, of course, be not a single source.
So what about the tumor volume of time?
Sorry?
What about the tumor volume over time?
Yeah, the tumor volume of time is not a full spectrum. So, yeah. But the tumor volume of time is indeed a variable metric to compare the predictions, but this does not necessarily indicate you when it's best to provide a follow-up data for 2023. It's just an indication of the tumor volume. That's it. It doesn't give you necessarily an indication about the vascular type. So our focus goes on the vascular. Because much plays out that drug oxygenation process, as well as the effectiveness of the therapy, which we do not touch with this slide. So the vascular is the, let's say, on the spotlight in those 6 studies. Yeah, so all up different, let's say, human scenario, that was assuming that we have this control means that you have been planted on the mouse, the tumor has grown, the vessels have reached this kind of pathological density. And then how about we run different scenarios where we have separately run, let's say, we have administered separately anti-angiogenic treatment. Moderate dose, low dose, or extremely high dose, which is, to be honest, on the far right, which is problematic. We don't like to administer a lot of anti-angiogenic treatment because this way you cause a lot of damage and process in uncertainty. So yeah, it's not good. So yeah, I hope I'm satisfied with those flows. You don't see that much those flows volume or like over here. See that this enhanced model that we have built will have different, let's say, phenotypes of the cancer cells. We can monitor exactly the amount of cells proliferative, hypoxic, and also quantify the process, which was not possible on the previous version of the model. And those are, let's say, some results, some results. So we consider low dose, middle dose, and high dose, and these directly address the vascular density, as shown on those networks on the top of the figures. While they still limited, so both of them show the tumor outline, which is those gray things, on week 7. And so several weeks after the implantation of the tumor on the mouse. And so by inspection of those bar plots on the right, we observe that tumor vascular density regression is strongly associated with final tumor volume. But unfortunately, as we destroy the vascular group, prevent the tumor from growing, we are also limiting nutrient supply with other tissues as well. Now, in the other treatment scenario of vascular normalization, we observe that causes substantial reduction in the distichial fluid pressure, which is attributed by many cancer biologists as supposed to be firewall or drug delivery. So it's kind of blocks the transport, the diffusion of the drug. Because it's pressure and accumulated in this tissue in successive amount. Okay. However, we also observe that at the same time, it is led to so the reduction of the fluid pressure reduction is led to improving the perfusion of the blood which is also a very important indication for cancer biologists. We'd like to reduce the vascular density, but we'd like to improve also the perfusion. Improving the perfusion means that you have an improved delivery of the drug through the venous system and so on. Well, more results. I'll kind of skip the discussion. These are simulations of metronome chemotherapy. So instead of a single dose injection, you can see on this plot, the smaller doses of equivalent cumulative dose to the single one. And we see here that the effect of the tumor development is more pronounced compared to the single dose treatment. While if we consider 5 smaller doses, 1/5 of the initial administration, the tumor seems to almost eradicate it. Also, we note here that the importance of not only looking at the effect of the tumor, but on the healthy tissue at the middle bar plot, the fractional. So this is the plot indicating the fraction of healthy tissue. We see that the metronomic chemotherapy decreases the magnitude of hypotheses, which is another important factor that especially radiation physicists consider. Pronounced hypoxia means reduced effectiveness of the therapy. And then I close this relatively large section with a nice, let's say, demo of the modeling on a mouse, using mouse data on this immunodeficient mouse, where instead of breast cancer cell being planted, colorectal cancer cells. So the particular cancer cell line, they also injected a vascular destructive agent and also another stainer, so as to indicate the vessels that are infused and so on. And we used information like scans, ex vivo scans of the network, micro-CT scans of the tumor from the mouse, and we fit them all together into the simulator to simulate either a small cytotoxic agent or liposome, this 5-fluorouracil drug. The first one, in principle, suppresses cell cancer cell growth, whereas the other one promotes cell death. Okay. So those drugs have a different effect, but it doesn't matter. Our, let's say, the take here was to predict the distribution of the drug. So how much drug what does it reach? So
is it the tumor there is before they die on this?
so yeah, so that particular information are cleared out a lot of things, just to make it visually appealing. So those things are vessels, and the more red they are, the more well perfused they run those simulations. And the contour is the drug concentration that has bound to the cancer. Cancer. Now, I have removed the periphery of the vascular network. There's no show in the background. That's because it's the health. So we're not focusing on that. Now, what you see here are tumor vessels. So the tumor-induced vessel, and the tumor is somewhere within, not smaller than that. I mean, it's almost as big as. Yeah, I have those measures from YouTube to take see them. And then some more exciting stuff. A couple of years ago, we started this collaboration with the German Oncology Center, where they are experts, truly cycles in brachytherapy treatment. So brachytherapy. So this is just a slide to explain what it is. is a technique for managing prostate. So for treatment, not prostate. So instead of external irrigation, this is internalized irrigation. So what they do is they stick those needles onto the pelvis of the male patient, and after those needles have been positioned properly, they infuse the drug. Okay? And over here is they just use some probe and they use it as a means to inspect while they put the needle in where the needle is positioned with respect to the gland and with respect to the urethra, which is part of the definitely not going to be part. Now, in principle, they put those needles in, but the problem is that on the one hand, the modality that they use, ultrasound, it's not the best one. It doesn't cooperate with the needles, meaning that as you can see on those images. Sorry, skip slide, but I just want to explain this a little bit better. So while those needles, metallic needles go in, they cause noise to the ultrasound. So they reflect the signal in principle. Yeah. Which means that the doctors may mistreat patients, meaning may not target exactly where they had originally planned, as over here. So what you see on the bottom right is simply the contour where they plan to insert the needles, and this is the contour that tells them, okay, this is going to be an effective distribution of the blood. Now, the problem on their side, the problem was how can we improve it in order to effectively improve it. And this where it comes for the engineering support. How can we build a model not predictably accurate and the right position of the needles, and how these needles will affect for the tumor and more importantly, the prostate. So Mira has been leading this project as part of her fellowship, and she used retrospective data in order to build, say, anatomically relevant, let's say, models, 3D models of the prostate, as you can see on the blue plot. So this is kind of an overlay of the model on a slice of an MRI scan. So we use MRI scans, scans, record scans altogether, as well as the information provided by the doctors in terms of the needles they provided, so they inserted. And the goal was to model how the prostate forms. Okay. After the insertion of first intended signal, just a small disclaimer, what they do is they first insert two needles just to one for the prostate, and then they insert the needles that have to be used for infusing the blood. So this is a published work. It's about to go out very, very soon. And these are just two, let's say, demonstrating comparisons between the red outline, which is the tumor, so the prostate, and the ultrasound, which is on the background, and the sky blue is the segmented prostate gland. Another example, which part of the University of Cyprus internal funding, was simulating radiation-induced pulmonary fibrosis. Very briefly, pulmonary fibrosis is kind of a side effect of the immune system in the lung. It could be caused by cell not known causes, that's idiopathic pulmonary fibrosis. It could be caused by radiation, and we have radiation-induced lung fibrosis, or it could be used by viruses. So in fact, cause this kind of scarring of the lung tissue. And but this could happen on lung cancer patients as well. So, in collaboration with the Cytopsychology Center, we have access to a cohort of patients who were diagnosed with primary lung cancer, and who were diagnosed with lung cancer, who were treated with radiotherapy, and they unfortunately developed lung fibrosis, despite they being given anti-fibrotic agents like computational. The problem with those patients is that fibrosis can be really fatal, diagnosed early. So again, our argument was, and we build a model that could in effect predict lung fibrosis. So, all right, so we'll be using medical image data, like CT scans, baseline follow-up scans. And we also use the therapy dosage marks from those patients that we built 3D models in the lab. So from previous, the small animal scale, which is the large scale of the human. We described the development of the disease with respect to radiation using mathematical expressions like partial differential equations in order to describe the tumor regression, the lung fibrosis, as well as the remodeling of the extracellular matrix of the lung stroma. Okay. And we are at the stage of validation, and we compare those very old results. Where we make this comparison in the in vivo and the in silico simulations on a cohort of 15 patients. Yeah, probably we could resume, we could have a chat about this. We're really keen on having this sort of optimization ML learning on this task. Yeah, I'll leave that for the moment. And then I'm going into a different route from the lung to the brain. So modeling cancer and progression and data simulation. So the next slides will try to simulate how we kind of the work we're doing in terms of simulating patient response to selected with surgery, as well as techniques for reconstruct clinical data from clinical images. So this is still ongoing. This project from the CCRI. And the concept of the project is to build a model, imaging and simulators, in order to predict the response of metastatic on brain cancer patients and their treatment to surgery, which is a very particular technique that we do in the oncology center here in Nicosia. And so in this case, we include only metastatic brain tumors coming from the lung, breast, and liver. And we are only building a model, but also imaging tools for segmentation and registration. But in terms of modeling, which is my thing, so to say, we want to use different, let's say, modalities, tier one, which is our contrast tool, diffusion data, etc., as well as the third plan data, build the 3D model of the brain tumor, the brain, the tumor, the healthy region, the tumor region, the necrosis, the edema, and then run it in a high-performance computer in order to simulate the response to the patient treatment. Apart from the modeling, I also mentioned about the imaging. So different steps to process the data and then come up into this mass as we have it here. Those are some preliminary results. And then let me go very fast because I'm in front now time into a different kind of branch, which is tools to reconstruct data. When we have not enough data, but we have data. So in this case, we want to build a tool set for MR elastography imaging data simulation. So probably not very familiar with what elastography means. So this is on the bottom, the top left. This is kind of the individual line down, the coil, the magnetic resonance imaging coil, and together with that, they've been applied this kind of so to say helmet. Has cushions, and those cushions apply, let's say, a vibrational force onto the individual's head. And this vibration causes vibration to the skin, to the tissue, and then this propagates into the brain and so on. And the scanner can capture the relative motion of the molecules that translates into wave speeds, and those wave speeds, given a dispersion relationship, can give you an estimate of the shear motions. So the concept of elastography is through this non-destructive approach, testing approach, can extract a measure of motions, stiffness. However, the challenge here is currently, we want to investigate this data estimation approach for starting tissue displacement and pressure flux in this in silico 3D braining model. Okay, when we have partial elastography, not 150 slides, but just a handful of slides. As part of this simulation algorithm, we partially available MRE measurements acquired on a few slides onto a specimen field defined on a three-dimensional 3D model, as you can see here. Right? And as an example, a typical wave image on the slides is shown on the bottom sketch, as you see here. And the mapping requires an anatomical data to construct a brain model. Okay? And it is based on assuming an underlying tissue model that is encoded in a set of partial spatial equations that I have, as I have on this slide. So data simulation was based on a time-dependent partial data methodology. I'll have to be honest with you, this methodology, so please bear with me. But I kind of directly on this very important paper that we study. Okay, so the Silico pipeline was validated using synthetic data. So this first version of our work, we didn't use patient data. So we want to probe the capacity of this data simulation algorithm by producing ourselves by solving the wave equation of synthetic data. And we simulated, as I write over here, for last kind of theory of physiological brain. So, like I said, physiological brain not human. At the bottom, I have the covariant equations with the solid fluid. So we assume the biphasic formulation, that's this low, and right, we have pressure, so the boundary. Now, well, as part of this work, we have this training manifold with uniform sampling, where we casted out 500, approximately 500 simulations, with each including a series of 40 times. Okay. So just imagine this is not a star problem, it's a transit problem. Okay. So we have a wave traveling, sure waves, pressure waves traveling to the brain. However, we focus on the shear waves, which is exactly what the MRE apparatus monitor. Yeah, on the right, I have some snapshot of the environmental solution. It is for a particular value of boson ratios, gas molecules, and their C mixture permeability factor. Several milliseconds after the first pulsation of the and over here, I'm trying to just demonstrate the reconstruction error, which we had to quantify once we validate our model, and we computed the reconstruction error only on the displacement field, which is exactly what the MRE device scan can acquire, also the pressure field, which is another indicator which we have claimed that it can be used as a biomarker of pathology. And I'm closing. So
What are the frequencies between these types of data?
in the order of hertz.
And are they, is it something that actually quantify the aspect of the cell, or is it completely
No, this is not purely physiological brain.
in general for as a scanning method? Would
Yeah.
there be any effect to
One of the select points is that having the patient through an MRI, through a large sequence of MRI scans, and also on top of that, MRI scans, it may take quite a bit of time. So how about we need to few very good ones, MRIs, okay, and use them to reconstruct the whole brain. This means there's some better now. Does that make sense? But like I said, it's only for the physiological brain. Now, as far as the frequency spectrum that this is applicable, it will be exactly the same that the MRE acquisition takes place, for which other investigators have converged that yes, the image they image well. I don't know if it's good. But also. Yeah. And the last section, closing or slide, is about cardiovascular modeling. So two different branches. One is about simulating cardiac mechanics, again led by Miriam. That was actually her work at King's, and she got a lot of knowledge from that field. So the important here is how can we kind of estimate stress. So while as in the previous examples, patients, individuals, whatever, they are scanned before, meaning that things are live, meaning that they are under conditions where things take place like stresses, like pressure, etc. And the same with the blood vessels, the same with the heart. So it's an organ that cannot stop. And while imaging, we're not entirely certain about the condition of the mechanics perspective of stress. That's really important when it comes to subsequently simulating how they have mechanical deformed. All times people were ignoring this fact, but nowadays it's the state of the art that has to be acknowledged. And this is with Mia's contribution, that is to say, okay, can we estimate particularly for the heart, and what's the best way to assume that this is the zero, the stress-free or zero pressure situation? Can we use that to estimate the stress? So this was the motivation behind that. So to run different simulations using different configurations as the reference one. Unfortunately, I think I'm almost running out of time, so we'll go super fast. But just summarizing some results that we have to acknowledge a few factors when estimating this zero pressure configuration, like adjacent tissue, like the rib cage, like the lungs, so on. So those were important factors that others have been not considered as studies in terms of cardiovascular simulation, and also important things like the blood flow, and how this affects predicting zero pressure configuration. I'm so sorry that I have to really go fast forward and go to the last section, which is Nicolas' fellowship using now, instead of Silico, also machine learning in terms of quantifying the important factors that can be used that can lead towards rupture of cerebral needs. Well, fortunately, the slide I prepared the aortic arch. We're not focusing on the aortic arch, we're focusing on the Willis circle. Okay? And the idea is, on the one hand, let's say, extract the geometric features of the blood vessels being inside. I think I do have a lot of the morphology of metrics as I have over here, at the size of the sac, the size of the lumen, different branches of the aneurysm, as well as hemodynamic metrics. So the first one, the morphometric physiological metrics, will come by post-processing, and the hemodynamic metrics like blood flow, pressure, pressure, etc., come from CFD simulations. Now, the idea is how can we process all those quantities all together, and speaking about, let's say, quantities, I'm talking about 15, 20 metrics, without having any clue about which is the predominant factor and rupture. And also, we're talking about some 500 patients. So doing all those processing using statistics, probably is not entirely clear because of the complexity of the disease. So in this case, we employ a decision tree algorithm in order to extract the different features and also indicate which is the best marker that can give us a best result of rupture. So those are published results. They're going to be submitted for analysis very soon. This year, I think I have to close this summary. I hope like a few of the examples I showed you in terms of simulating tumor growth, angiogenesis, drug delivery, needle insertion, cardiovascular, disease progression in fibrosis, etc. A lot of things going on. I hope the message was clear in terms of what we've been developing and our capacity in terms of simulations. Like I said before, the Silico has been informed and validated using a large array of data, differently, not only in vitro but also in vivo, as well as in ex vivo. Special thanks to all the people in the group currently, also former members, last year's tough times, funders, and yeah, I would like to thank you. It's just a slide. The grant we received just a couple of months ago with the European Council, and we aim for going from simple in vitro experiments into in vivo experiments to something with effect of plasma in cancer. And this is a consortium between the University of Cyprus, will be leading, so one, Patterson and SMU in London. And yeah, we'll be having opening soon people who are experts in high-performance computing and modeling are very welcome to join the team. But I will keep you posted this. And again, thank you very much.
Very nice, Vasily. By reading your abstract and listening to introduction, I thought maybe at the end you are going to show combination of cancer and cardiovascular problems, especially both of these are observed at later age. But I guess maybe that's a question of your grandchildren or something, or your children or your students. But that's a kind of introductory joke, yeah? Unless you want to comment on it.
No, yeah.